In this first lecture of Module 8, we will take a closer look at the defining characteristics of the important invertebrates in the fossil record and what they tell us about their paleoenvironments. We will begin with four phyla: the Porifera, Cnidaria, Brachiopoda, and Bryozoa. But first, there are seven phyla and three classes of invertebrates important to paleontology and Earth history. These are phylum Porifera, Cnidaria, Brachiopoda, Bryozoa, Echinodermata, Arthropoda, and Mollusca, plus three classes of mollusca, the bivalvia, gastropoda, and cephalopoda. These invertebrates are important because they either have hard parts, are important indicators of paleoenvironments, or both. Many of them are used today as index fossils. We have talked a bit about phylum Porifera because they were some of the first metazoans to appear in the Precambrian. These photos are of three modern sponges in the oceans today. The basic characteristics of sponges include a porous structure with a body plan organized at the cellular level. They lack symmetry, differentiated tissues, and organs. The cells can switch functions from food gathering to reproduction, and they reproduce by both asexual and sexual methods. There are over 10,000 species, all of which are aquatic, and most of those are marine. The archaeocyathids are a special problem in paleontology. They were a Cambrian reef-building organism that evolved and went extinct in the Cambrian. They were an evolutionary dead end because there is nothing like them today. They were similar to sponges, but still very different. They had a structure that is characterized by a double wall. We do know they were filter feeders like sponges. The left diagram is the structure of an archaeocyatid. Water went in through the side and was expelled through the central cone. This animal was far more structured and compartmented than a typical sponge. On the right, you can see a fossilized Cambrian reef built by archaeocyathids. The archaeocyathids, this extinct group of possible sponges, dominated the reefs of the Cambrian seas. The image on the left is a cross-section of the animal, while the right image shows the external morphology of several of these cone-shaped animals. In the left image, you can see the double rings preserved very well. We have also talked briefly about the next phylum as one of the earliest metazoans. Phylum Cnidaria includes corals, jellyfish, sea fans, anemones, and sea pens. This photo, the photos here show modern examples of each of these cnidarians. The Cnidaria, or nettle bearers, includes jellyfish, sea anemones, and corals. Their cells are organized into relatively few tissue types with no specialized organization. They all have radial symmetry and specialized stinging cells called cnidoblasts. Most species are marine, although the hydrozoans can be abundant in freshwater. Cnidarians have two basic life strategies: the polyp and the medusa. The polyp life strategy is usually sessile, though some can jump and somersault across the sea floor. The medusa life strategy is a mobile pelagic life strategy. Polyps and medusae have the same basic body plan, but inverted. Look at the medusa in the diagram. Now, imagine it turned upside down. It looks like one of the individual polyps in the diagram. In this slide, you can see the three main groups of corals that have evolved since the Cambrian. The first on the left is a rugose coral, often called a horn coral. These were mostly solitary polyps. The tabulate corals, like the one in the center, were colonial polyps. Both the rugosa and tabulata are extinct today. All modern corals are scleractinians, which can be either solitary or colonial. Paleozoic corals were not particularly successful reef builders. Most Paleozoic reefs were built by sponges. Scleractinian corals expanded rapidly during the mid-Triassic and replaced poriferans as the primary reef builders. Scleractinians are corals that have a symbiotic relationship with zooanthely, an alga. This is one possibility for their rapid expansion. The next phylum is phylum Brachiopoda, one of the Lophotrochozoan group of bilaterian metazoans. As you can see here in the top middle photo, brachiopods are still around today. Brachiopods are twin-valved shellfish with a lophophore. Their valves, or shell halves, can be organophosphatic or made of carbonate. The brachiopod lophophore is a filamentous feeding organ. Brachiopods dominated the filter-feeding benthos of the Paleozoic, but never really fully recovered from the end Permian mass extinction. Living brachiopods are relatively rare. There are only about 120 genera that survive today. The phylum is entirely marine, occupying habitats from the intertidal zone to abyssal basins. They are bilaterally symmetric with two valves, the pedicle valve and the brachial valve, which open and close during feeding cycles. Brachiopod fossils are excellent for use in regional biostratigraphic correlations, and some forms have the potential for international correlation. Living brachiopods occur most commonly in higher latitudes, though some very delicate forms with organophosphatic shells reside in tropical waters. The soft parts of the animal are enclosed by two morphologically different valves. Brachiopods are bilaterally symmetrical, but the plane of symmetry is perpendicular to the commissure, which is the opening. The plane is along which is the plane along which the valves open, and the plane of symmetry bisects both valves. Larger brachiopod species with thicker, more robust shells are generally found at higher latitudes and at depth. For example, the fjords around Canada and Scandinavia and around Antarctica and New Zealand. Small to microscopic species with more delicate shells are typically found in the shallow tropics, for example, in reef environments where they are protected. The second phylum, belonging to the Lophotrochozoan group of bilaterian metazoans, are the Bryozoa. Three of these photos show modern living bryozoan colonies. The far right picture is a microphotograph of a fossilized colony. The Bryozoa are the only phylum in which all species are colonial. The colonies are made up of tiny individuals called zooids. There are approximately 16,000 fossil species and 6,000 living species of bryozoa, and most are marine, like the brachiopods. Bryozoa are also lophophorates, but in contrast to brachiopods with a filamentous lophophore, the bryozoa have a tentacular lophophore with anywhere from 8 to 100 tentacles. Zooids are enclosed in an exoskeletal chamber called zooetia, which is composed of gelatinous, leathery, or calcareous materials. The primary function of zooids is to capture food, but some specialize in defense, reproduction, or sediment removal. The lophophore extends outward from the zooid and directs food to a central mouth that leads to a U-shaped gut. Each individual zooid is a hermaphrodite containing both male and female reproductive organs. In this illustration, you can see how each zooid is attached to a stolon and how the lophophore is very different from the brachiopod lophophore. The majority of bryozoans are part of the sessile benthos, ranging from the sublittoral zone to the edge of the shelf. Some can even be found at depths greater than 1 kilometer. They are sensitive to environmental parameters of temperature, depth, salinity, substrate types, and turbulence. The shape of the colonies depends on environmental conditions. As such, they are good indicators of paleo environments. Shallow water colonies like these are and were dominated by encrusting, thick, erect, or rooted and free-living forms. These thick forms are indicative of a high-energy environment. The top photo is an erect form, while the bottom is an encrusting colony. Deeper water environments are occupied by mainly attached and rooted forms. They are often delicately branching, which indicates a low-energy environment. On the top, you can see many delicate deep-water bryozoan fossils, likely similar to the modern bryozoan in the bottom photo. This is the end of part 1. In part 2, we will explore the characteristics of phyla Echinodermata and Arthropoda.